Good morning, everyone. This is Ankita from MedGenome, and today I will be presenting on an accurate prediction of neoantigen vaccine candidates using OncopeptVAC and OncoPeptSCRN. As a reminder, we will be taking questions at the end of the presentation, and if we run out of time, we will get back to you via email with your questions. So moving along with the agenda for today, I will briefly review the opportunities and challenges currently in cancer immunotherapy. I will then discuss the importance of neoantigens in cancer immunotherapy, then review the challenges of accurately predicting immunogenic neoantigens. I will then highlight MedGenome's OncopeptVAC neoepitope prediction pipeline and OncoPeptSCRN assay for accurately validating neoepitopes. And I will then present a case study of how we've used our OncopeptVAC and SCRN solutions to predict neoantigen vaccines for therapy in Lynch syndrome. And I will end this presentation with summary and conclusions. This graph here is an overview of the current modalities of treatment in cancer immunotherapy, and on the x-axis you can see the different types of cancer immunotherapy regimens that are in place, such as checkpoint inhibitors and other immune modulators, vaccines of interest, oncolytic viruses, as well as bispecific antibodies. What is interesting to note here is that while there are several ongoing clinical trials as well as preclinical discovery for each of these regimens, there are only very few approved drugs that are approved for cancer immunotherapy. And why is that? So in this slide here, I'm showing data from Shalpa et al.'s review, where you can see that of all the checkpoint inhibitors that are currently being used, there's only 30% of patients are approved to treat with that inhibitor. Furthermore, if you look at the long-term benefits of all cancer treatments of the checkpoint inhibitor drug, you see only 8% of patients have long-term benefit. And the reason for this is in a lot of cases you have a lack of good predictive and prognostic biomarkers as well as biomarkers of early response to treatment. While there are several challenges in the field of checkpoint inhibitor therapy, there are strategies that are being put in place to improve the efficacy of these inhibitors. And these strategies can be categorized into three broad buckets. One of them is to break the tumor tolerance. The other one is to combine therapies with other checkpoint inhibitors. And the third is to improve and discover new biomarkers for better patient selection and response prediction. So under the category of breaking tumor tolerance, one can use personalized vaccines or shared vaccines. One can also combine checkpoint inhibitors with radiation to improve the inflammation and the cancer. One can also target inhibitory cell types, immune cell types that are present in the tumor microenvironment, and target anti-angiogenic pathways to improve delivery of drugs. And lastly, and this is an emerging area, one can manipulate the patient microbiome to achieve superior response. Now, in the combination therapy space, there are several combination therapies that are ongoing in clinical trials that have showed improved efficacy of checkpoint inhibitors. Now, with biomarkers, in order to identify biomarkers, what has been shown to be important is the immunogenicity of a tumor, such as measured by the neoantigen load, and the tumor microenvironment, such as the presence of various immune cell types beyond the CD8 positive cells that are targeted for immunotherapy. So by identifying new biomarkers and looking at the tumor microenvironment and looking at factors that can reduce immune-related adverse toxicity, we can improve the efficacy of checkpoint inhibitors. In today's presentation, I will briefly review how at MedGenome, using the OncopeptVAC solutions, we are able to predict and develop personalized and shared cancer vaccines I will also show some evidence of how our solutions can be used to combine and predict, come up with combination therapy solutions, as well as identify inhibitory cell types that can then be targeted. And we will present on how we identify the immunogenicity in the tumor and look at the tumor microenvironment. So for the next few slides, what I will focus on is how we have utilized our OncopeptVAC solution at MedGenome to identify personalized vaccine candidates. Now shown here is a schematic or an overview that goes into the general workflow of how neoantigen candidates are predicted and validated currently. So one can start with a patient tumor sample and perform whole exome sequencing of the tumor and the matched normal to identify mutations that can lead to neoantigens, and then use RNA-seq to identify the neoantigens that are expressed in the tumor. After that's done, these candidate neoantigens are then fed through a neoepitope prediction algorithm that in most cases uses HLA-binding affinity to find potential immunogenic neoepitopes. Now, these predicted neoepitopes are then screened via a cell-based assay which looks at the activation of cytotoxic T cells by interferon gamma and other markers of response. And then these candidates are then injected back into the patient to look at vaccines. In order for a neoepitope to be immunogenic, not only does it have to be presented correctly on the antigen-presenting molecule and so be bound by HLA, it also needs to be recognized by the TCR and activated by the TCR. Now, since these two steps are so critical, I will briefly review the steps that have to happen for accurate new MHC binding and presentation, as well as TCR recognition and activation. So this cartoon here summarizes the cellular steps that happen for an antigen to be processed. So these antigens are fed through the proteolytic machinery and undergoes cleavage and processing. It's then trafficked to the ER, where it gets loaded onto the MHC molecule and packaged into vesicles that are then presented on the surface of a cell, of an antigen-presenting cell. And then that can be found by the TCR. Now, in order for a peptide to be presented or be recognized by a TCR, I'm showing you a cartoon here of what happens. So this is an MHC-bound peptide, and you can see here that there is a bend or a turn that the peptide, going back one slide. I apologize, there is a little delay in the So as I was saying here, in order for this peptide to be recognized by the TCR, it takes a turn for it to then bind the TCR binding groove. And in fact, this property of the peptide is critical in determining whether it's going to function as an immunogenic peptide or not. So in summary, in this slide, what I've shown you is that for a new antigenic vaccine candidate to work, it needs to get processed into peptides via the MHC binding and presentation pathway and has to be found by the TCR, telling us that all of these properties need to be looked at when trying to predict accurately neoantigens that will be immunogenic neoepitopes. In this slide here, I'm showing you what is one of the current methods that is utilized to predict neoantigenic epitopes. So what is shown here is a graph that shows the HLA binding affinity of immunogenic and non-immunogenic peptides. And this dotted line here represents the cutoff below which all peptides have an binding affinity of less than 500 nanomolar are going to be called immunogenic. Now, using this method, if you look at simply the immunogenic peptides here on the right, what you see is there are several peptides that are being called as immunogenic. So these are true positives. But indeed, there are ones that are based on the HLA binding affinity being classified as false negative, but in fact, they could be positive. Now, if you just go back here and look at the non-immunogenic peptides, and again take the same criteria of HLA binding below 500 nanomolar, what you now see is that these non-immunogenic peptides indeed are being called as immunogenic, hence they're all falsely being identified as immunogenic, versus only a subset are being called as true negative. So in summary, what I'm showing you here is that using this HLA binding affinity-based algorithm, you only predict 60% true positives and 40% true negatives, which means that there is a need to develop a method that is more accurate in predicting immunogenic versus non-immunogenic peptides. So this slide here is showing you the distribution of amino acids in the neoantigen peptides of positions 1 to 9 and the amino acid sequence of both immunogenic and non-immunogenic peptides. So what you see here is at position 2 and position 9 is where the amino acids bind to the HLA molecule, and here there is a high frequency of occurrence of hydrophobic amino acid residues. So in fact, by looking at the amino acid sequence of a peptide, you can determine that these peptides can potentially bind HLA. Now, if you look at positions 3 to 7, these are the anchor residues for binding to the TCR, but in fact, there is no consensus amino acid sequence that is enriched preferentially in these regions, telling you that for determining TCR binding, looking at the amino acid sequence is not sufficient. So another criteria that has been utilized to look at or to assess immunogenicity is hydrophobicity of the peptides. And what you see here is if we look at the overall hydrophobicity of the 9-mer peptide for immunogenic and non-immunogenic, you really don't see a much difference in the average hydrophobicity, telling us that neither looking at the amino acid sequence or the hydrophobicity is sufficient to explain the immunogenicity of neoepitopes. So at MedGenome, what we then did is to take a broader approach where we've developed the OncopeptVAC pipeline using machine learning, where we not only look at the amino acid sequences, but also all the physiochemical properties of amino acids. And I'll walk you through how we built our pipeline. So the pipeline was built in 3 steps. First, we selected training data to build a library of peptides that would then be utilized for building the algorithm. Then we generated features that, again, include all the physiochemical properties of amino acids, and then we validated our models. So to build the training data, we took immune epitope database data sets of over 1,655 peptides, and then we narrowed it down or filtered them based on the ones that had biological assays for immunogenicity, and only selected immunogenic peptides that were of HLA class 2. And with that, we came up with 423 unique peptides, of which 307 were classified as immunogenic and 116 non-immunogenic based on assays. We then utilized several softwares and algorithms to build or compile all the features that would then be utilized to build our machine learning models. And then we input some training data sets from the IADB data to then validate the models that we were building. So here, what I'm showing you is a graph that goes after the performance of the machine learning model that we built using all the features from the test data set. And in fact, what you can see is if we utilize all of the physiochemical properties, we get models that have very high false positive rate, telling us that we needed to improve our prediction. So then we went back to our models or our algorithm, and we did feature reduction by selecting features and models that were only using a voting method that were only represented over 50% of the times or over 50% times calling positive versus negative. Now using that, what you can see here by this dotted line is that we've significantly improved our sensitivity and specificity in detecting immunogenic or calling immunogenic versus non-immunogenic peptides. So finally, using several of these models, we've built a classifier and the OncopeptVAC pipeline which gives the user an immunogenicity prediction score, where a score of less than 0.5 calls a peptide as non-immunogenic and over 0.5 is classified as immunogenic. So in the next few slides, I want to show you what did we find using what did we find about the features that help to define immunogenicity versus non-immunogenicity. So before I go into that, I just want to remind again that if you look at the structure of this MHC peptide TCR complexes, which is also shown here by the crystal structure, and here the magenta represents the peptide, what you see is that amino acids 1 and 2 of the peptide are what is interacting with the MHC molecule and has hydrophobic residues adjacent to it. But in fact, at position 3, indicated by this arrow, this peptide takes a turn, which then allows you to go and interact with the TCR. So in fact, there are hydrophobic residues or hydrophobic interactions happening, and this turn is very critical for, that's what the literature tells you. Now here is a heat map summarizing the top features that were selected by OncopeptVAC that was utilized to call immunogenic versus non-immunogenic peptides. So the x-axis here is showing you each position in the peptide, and the y-axis are the top features that were selected. And the colors here, the dark blue, represents the number of times these features are found in each position of the amino acid. So the way to interpret this data is that if you look at position 2 and 3, you see that there is an enrichment of several features in these amino acids that pertain to helix-turn helix or a helix turn. And that's what I just showed you, that at these positions, the peptide needs to take a turn to go and interact successfully with the TCR and potentially mount an immune response. And what you also see, for example, is that there is, if you look at beta sheets, essentially at all these positions, there is no propensity of finding a beta sheet, and in fact, that's what we see. So taken together from this data, what I've shown you is that the features predicted to be important using the OncopeptVAC pipeline is in fact what is found in the literature if you look at crystal structures of the MHC peptide TCR complex. So in summary, using the OncopeptVAC solution compared to the net MHC cons, which uses HLA binding affinity to predict immunogenicity, you see that we've improved the sensitivity of detection to over 90% and the specificity to over 99%, telling us that OncopeptVAC is a more sensitive and accurate method to predict immunogenic neoepitopes. So now I want to move on to discussing our other assay, the OncoPeptSCRN, which is the assay that we use to validate ex vivo the candidates that are predicted using the OncopeptVAC. So in this is again a reminder of how this works. So we have taken from NGS data, we take express neoantigen candidates of 9 more peptides, and then we feed it through our OncopeptVAC pipeline shown here in this box. So as you can see here in the OncopeptVAC, we're taking pretty much all the properties that are required for a peptide to be processed and presented, but we prioritize using the TCR binding. That's why it's on the top. And once we have the candidates, then we feed it through the OncopeptSCRN. And so what is the OncopeptSCRN assay? In the OncopeptSCRN assay, essentially we are assaying for the functional CTL response or T cell response of CD8 positive T cells. And to deliver the peptides of interest, we either add it directly to the media or clone it into a mini-gene delivery method. And again, we assay using FACS and other NGS methods. So just to give a little bit more detail on how the OncopeptSCRN assay is set up, you can start with either healthy donor or patient PBMCs. And there are two modalities. One is the high-throughput screen, where you then add in cytokines and peptides at day 0 and continue to culture the cells. These PBMCs have both the antigen-presenting cells as well as the T cells that will get activated upon binding to the peptide. And then we assay at day 22 for T cell activation by looking at interferon gamma signal in CD8 positive T cells, which is shown by FACS here. Now, while this high throughput method is very convenient in quickly screening several peptides, we recognize that since we're adding the peptides exogenously, the endogenous processing that has to happen in a biological condition is not necessarily getting recapitulated. So for a more thorough analysis, we have developed a mini-gene-based peptide screen, wherein, in this case, we take PBMCs and we sort out monocytes or immature monocytes that will later be antigen-presenting cells from the T cells, and then we electroporate peptide-containing mini-gene into these dendritic cells, sort out the ones for maturation, and then co-culture with the T cells and assay for response using the same method shown here. So what I've shown you here is that there are two different methods that we are using to screen our peptides. Now, both of these options are available through MedGenome as a service if you're interested. So now I want to show some proof of concept validation data of OncoPeptSCRN. So in this assay, first I want to show the high throughput OncoPeptSCRN assay data. So what we've done is we've taken peptides that were identified from the literature to be immunogenic from some cancers, and then we add in the wild type and mutant version with the DMSO vehicle control. And shown here is the response of interferon gamma in the CD8 T cells, and what you can see is that compared to the DMSO control, in the peptide of interest, there is over a 2% upregulation of interferon gamma, and this is melon A, which is a positive control peptide, telling us that our assay is working. Now, using this method, currently to date, we have screened over 96 peptides that were predicted to be immunogenic by OncopeptVAC, and we find that over 81% of these peptides are positive based on our screen. And then for non-immunogenic, we started out with over 9, and we've seen that over 7 of them are negative, telling us that in combination with the assay, we have over 80% accuracy in predicting immunogenic versus non-immunogenic peptides. I now want to move on to showing some data of how we have validated our mini-gene platform using the OncopeptSCRN. In this, again, we are trying to screen for cancer neoepitopes and, again, do a similar assay to look for immunogenicity in CD8 positive T cells. And in this case, we've used constructs of just the vector alone or a mutant peptide. I should mention that because this is a frameshift mutation that we're screening, we don't have a wild-type counterpart. And then again, we did a flow cytometry analysis. So what you can see here again on the left is the CD8 T cells or interferon gamma in CD8 T cells that were exposed to an antigen-presenting cells which has an empty vector versus a mutant peptide. And in fact, compared to the empty vector, in the mutant peptide, you have over 2.5% upregulation of interferon gamma, telling us that, in fact, this mutant peptide is immunogenic and can be utilized or screened further or developed into a cancer vaccine. Now I want to move on to a patient case study of Lynch syndrome, where we've utilized our OncopeptVAC and screen solution to identify neoantigen vaccines. And just to give a little bit of background about Lynch syndrome. So shown on the top is an image of a normal colon, and what happens is in certain individuals you have these colon polyps which are benign, but upon further mutations, a subset of them develop into carcinoma. Now, in a typical population, the rate of progression from a benign polyp to a carcinoma or an adenocarcinoma is 10 to 15 years. But if you have these mutations mentioned here, there is an accelerated progression into adenocarcinoma. And more importantly, if you look at the incidence of cancer in the U.S. of individuals carrying these mutations, you see that there is an over 80% risk of having a colon cancer. And so this is a real unmet need in the community to develop some immunotherapy drugs. So what we then did is at MedGenome, we took a MSI-high phase 3 cancer patient sample tumor, and we decided to look for immunogenic peptides. And so what's shown you here is we did sequencing, and we ran through OncopeptVAC and found a subset of these mutations in both missense and indels that were predicted to be immunogenic. And in order to prioritize the candidates that we would screen further in our assay, what we did is we went back to the literature and picked up these peptides that were thought to be important in the biology of the development of the cancer. In fact, that's shown here is that they have a high, they're predicted to be positive, and they have a high frequency of occurrence in the general population of all the somatic variants that are found in colon cancer. Now, before we did our screen, as I mentioned, we wanted to make sure that this neoantigen is actually expressed. So we did both whole exome sequencing, and we see that these candidates, the mutant allele is there in a subset of the cells from the tumor. And in fact, if you look at RNA-seq, while very low in expression, you do see that a subset of the cells in the tumor have expression of the neoantigen candidates. So we then decided to proceed to do the screen. Now, before we screened so to do the screen, we utilized PBMCs, both from the healthy donor and the Lynch syndrome patient. And what you see here is the result from the healthy donor. And again, we are looking at the level of interferon gamma in CD8 positive T cells. And in fact, compared to the wild-type peptides, in the mutant peptides, you do see an immunogenic response. Now, if we look at what's happening in the patient, what you see is for the 3 candidates, there is still immunogenicity, but for AXIN 2, there seems to be a loss of immunogenicity, telling us that this patient has been tolerized to this mutation. In fact, if we were to make a vaccine used for this particular candidate, it probably wouldn't work. So now, in order to really come up with a therapy that would work, of course, based on what I've told you so far, we have several antigen candidates that can be packaged into a cocktail and delivered as a vaccine. But just to look at the bigger picture here, a tumor has the microenvironment, which I'm showing you here in this cartoon, and it not only secretes these neoantigens that can then be targeted by CTLs, but in fact, in the tumor microenvironment, there are these other immune cell types, such as the dendritic cells and macrophages, that can inhibit immune response. So for us to determine whether this Lynch syndrome patient would in fact respond to immunotherapy, we decided to look at the tumor microenvironment. And for that, we used our other solution, the OncopeptVAC, which is our tumor microenvironment analysis pipeline and uses a proprietary minimal gene expression signature of all immune cells and scores for the relative enrichment of all immune cells in the tumor. So again, in this patient sample, what you see from this in this tumor is that we do have CD8 expression, which means that checkpoint inhibitors will work, as well as Tregs, but there is a very high frequency of occurrence of these inhibitory cell types, such as the MDSCs. And so we went back to the literature, and this is our proposal for a combination therapy along with the vaccine that would now, again, lyse the inhibitory cells and create a more immunogenic tumor environment. So in summary, what I've presented to you today is MedGenome's OncopeptVAC solution, which can be utilized to predict therapeutic cancer vaccines. And I've also shown you some evidence of how we can use our OncopeptVAC, which is the tumor microenvironment analysis solution, to fine-tune combination therapies and predict biomarkers of response. In addition to just predicting therapeutic targets, at MedGenome, we also have the OncoPeptSCR solution, which allows one to look at the TCR repertoire before and after treatment and do immune monitoring, and in fact, use TCRs as a biomarker of response. So the TCR profiling we offer as a service as well, along with OncopeptVAC and TUME, and we are using 3 commercially available platforms that we have implemented into our workflow, along with NGS analysis. So if you're interested in learning more about our services, please visit medgenome.com or contact us at research@medgenome.com. So in summary today, what I've shown you is that OncopeptVAC is a more sensitive and accurate method for prediction of immunogenic new epitopes, and this utilizes extensive features that are relevant for proper presentation and activation of T-cell response. I've also shown you the OncoPeptSCRN platform that can assess immunogenicity with high sensitivity and accuracy. And lastly, I've shown you how we've combined OncopeptVAC along with our tumor microenvironment solution to design potential combination therapies and understand mechanisms of resistance. With that, I would like to thank everyone for attending, and I think we can move. I will be taking some questions. So we have a question here that to generate training data, did you run HLA-A2 or just HLA, more than just, did you just run HLA-A201 or more than HLA-A201? Yes, so the answer to this question is that to generate our training data, or to validate our model for that matter, we have only utilized HLA-A201. And the reason for that is in the IADP database, if you look at ambiguous or non-ambiguous peptides that are considered as immunogenic, majority of them are in that category. But just to qualify this point, when we do our screens, we are testing it in patients with multiple HLA alleles. So the next question is, how does your algorithm differ when predicting binding affinity versus something like MHC NetPan? So in answer to this question, in our algorithm, we also consider HLA binding as a criteria because it's needed, but that's not how we prioritize. We then look at the ones that are TCR binding and then call out positives. So the next question is, how does your prediction system for HLA types go beyond standard Caucasian alleles? So to look at this, we are working in collaboration to have access to genomic data from individuals outside of the Caucasian population to then look at exome sequences and find variants. So I think we have run out of time for this webinar. So I will, and I see there are other questions that have come in. So in response to that, we will follow up with an email. And thank you all for attending.